These notes are meant to help you complete your lab, the Intro to Gregor Mendel and Mendelian Genetics. And really this is a focus on how to set up and carry out your Punnett square analyses. So before we begin, I want to do a little bit of review on some of the vocabulary that we've been learning. So recall with Mendel's experiments on pea plants, he used what he referred to as the parental generation, which you see here. Mendel called these true breeding, but we now refer to these as homozygous individuals. This word homozygous means that their genotype has the same alleles. So if we were to look at this purple flowered parent and look at their genotype, both of the alleles of that genotype would be the dominant P, the capital P. If we were to look at the white flowered parent, both of their alleles are the lowercase p. So the two alleles that make up the genotype are the same. So that's why we refer to them as homozygous. Then the offspring of the parental generation is referred to as the F1 generation. So here's the F1 generation. It stands for first filial. Mendel referred to these individuals as hybrids, but we now use the term heterozygous. That prefix hetero means that the two alleles are different. So if we were to look at that purple flowered plant, it has one dominant P and one recessive P, so a capital P and a lowercase p. And then if we allowed that F1 generation to self-fertilize or cross with itself, that's how we produce the F2 generation, or the second filial generation. And this is usually the generation that we ask about when we're asking about genotype frequency or phenotype frequency. So because the purple flower trait completely masks the white flower trait, remember we call that the law of dominance. So a dominant allele will hide the presence of a recessive allele. So this is why we call the purple trait dominant, the recessive trait, the white trait recessive, and we use a capital letter for the dominant and lowercase letter for the recessive. So anytime we want to indicate a purple allele, we would use big P for purple, and we would use little p for white. So this box is just showing you all of the possible allele combinations that we can make. We could have homozygous purple, which means it has the two purple alleles that are the same. We could have homozygous white, which means it has the two white alleles that are the same. Or we could have heterozygous, which would be purple because it has that capital P, which would hide the lowercase p and it would look purple. Right. Lastly, we use these terms genotype and phenotype. Remember that genotype refers to the letters used as symbols for the genes. So a genotype is always going to look like big P, or big P, or little p. All right, so it's that combination of letters. Whereas the phenotype is the appearance. So for instance, we were using the appearance of having purple flowers or the appearance of having white flowers. It should always be a description of what the organism looks like. So let's use a slightly different trait. Let's look at round versus wrinkled seeds. When Mendel crossed his pea plants that were round and wrinkled, he determined that the round trait was dominant and the wrinkled trait was recessive. So if we look at these genotypes, if an individual has two dominant alleles, then they will have the dominant trait, which is round. Even if they only have one dominant allele, that's enough to hide the recessive, so it would still look round. And if there were no dominant alleles present but only two recessive alleles, then it would be able to show the recessive trait, which is wrinkled. Right? So let's practice assigning letters now. Like if we assigned a genotype, remember that in order to have the dominant trait, you could either have two dominant alleles. So this is what we would call homozygous, dominant. Or you could just have one dominant allele and one recessive. That one dominant allele is enough to hide the recessive, so it would still look tall, and we would call this the heterozygous genotype. Right? If there were no tall alleles present, only short alleles, then the individual would take on the short trait, and we could still call this a homozygous genotype because the two alleles are the same. So if we look at a sample organism like guinea pigs, their hair length is a genetic trait where short hair is dominant to long hair. So if we use an H for the trait, if we had two dominant alleles, then the guinea pigs would display the dominant phenotype, which is to have short hair. If the guinea pigs were to display the heterozygous genotype, big H, little h, that big H is still going to follow the law of dominance and cause the organism to display short hair. If the organism had the homozygous recessive genotype with two little h alleles, then it would display the recessive gene, which is long hair. So let's use this to help us figure out how to set up a Punnett square. Your rules for setting up a Punnett square are right here at the top, and these rules will never change. So we always use it the same way. We're going to establish the genotype of each parent, place one parent genotype across the top of the Punnett square, the other parent's genotype on the side of the Punnett square, and then we're going to fill the Punnett square in. So let's show a cross between an individual who is round, big R, and wrinkled, big R, little r. We place the first parent's genotypes along the side of the Punnett square. You can see that here. The second parent's genotypes go across the top of the Punnett square. You can see that here. And then to fill out the Punnett square, all we do is we carry the alleles from the side over into any box that's next to it. So that would be this box and this one. We'll do the same thing for this allele down here. We'll carry it into this box and this box because they're to the right of that allele. And then we'll carry the alleles that are at the top down into the boxes that are underneath them. And this little r would be carried down into this box and this box. So that shows us all of the possible offspring outcomes. So then we can answer a question, what are the phenotypes? So how many out of 4 would be round? So we would have to look into this Punnett square and look for that dominant allele. This box has dominant, so it would be round. This box has a dominant allele, so it would be round. This box has that capital R, so it would be round. And so does this one. So all 4 of our possible offspring outcomes would be expected to be round, and therefore 0 would be expected to be wrinkled. Let's try another one. Now we're going to cross 2 heterozygous parents. So again, we're going to take this parent's alleles and write them at the side of our Punnett square. The second parent's alleles we're going to write across the top. Then we're going to carry those alleles into the box. This R goes into the box next to it. This R goes into the box next to it. Then we're going to bring this big R down into the boxes underneath it. And we're going to follow the law of dominance, which states that we always write a heterozygous genotype with the dominant allele first. We would never write it like this. Okay, that goes against the law of dominance. And then we'll carry this little r down into its boxes. So now, how many out of 4 would be round? Look for the dominant alleles. This box has 2 dominant alleles, so it would be round. This box has 1, so it would be round. This box has 1, so it would be round. But the last box doesn't have a dominant allele, so only 3 out of 4 of these would be expected to be round. 1 out of 4 would be wrinkled. So this is where we get the genotypic or phenotypic ratios. In this case, 3 to 1 would be a phenotypic ratio, 3 round, to 1 wrinkled. If we wanted to do a genotypic ratio, we had 1 big R, we had 2 big R, and we had 1 little r. So this is how we build our ratios. Okay, in the next cross, we're going to take a round parent who is big R. So we've got big R, and we're going to cross that with a wrinkled plant. So what would the genotype have to be in order for a plant to be wrinkled? They would have to be little r. So we'll use those genotypes to fill out our Punnett square. The first parent's genes go across the side. The second parent's genes go across the top. And then we'll fill that Punnett square out. We'll bring the big R over and the little r over. And then we'll drop the little r down into the box and the little r down into the box. So now, how many would be expected to be round? Hopefully you said 2 out of 4, these 2. Which means the other 2 that are little r would be wrinkled. We can also express these as percentages. So if I asked you what percentage are round, since we know that 2 out of 4 are round, you would divide 2 divided by 4, which would give you 0.5, and then you multiply that by 100%. So we would say that 50% are round. Right, let's do just a few more crosses. Consider a cross between a heterozygous yellow seed and a homozygous green seed. Right, well first we would need to remember that yellow is dominant. So if we have homozygous big Y, it will produce a yellow phenotype. If we have the heterozygous genotype, it will still be yellow. But if we have little y, that's what produces green. So now let's go back to our question. Heterozygous yellow, what is that genotype? Hopefully you said that heterozygous yellow is big Y, little y. And then in order to be crossed by a green seed, what would the green genotype be? This should be little y, little y. So those are our parent genotypes. We use them to fill out the side of our Punnett square and the top. And then we carry those alleles over into our boxes and carry the alleles from the top down into the boxes. So how many of these offspring are expected to be yellow? Hopefully you said 2. The top 2 would be yellow, which means that the bottom 2 would be green. All right, now let's try this one. Homozygous yellow is crossed with a green seed. Hopefully you said homozygous yellow is big Y, big Y. Green would have to be little y since it's recessive. So we put big Y on the side, little y across the top. We carry our big Y's over into the boxes next to them, carry our little y's down into the boxes underneath them. And how many of these are expected to be yellow? Hopefully you said all 4, since all 4 of those boxes contain a dominant allele. So none of them are expected to be green. Now for your lab, you're going to have to complete these without me giving you the steps. So you'll have to know your first step is to find the genotypes of your parents, then to fill out your Punnett square with those genotypes, carry your alleles into your Punnett square to fill it out, and then use your Punnett square to answer the question. So the question is, what percentage of the offspring will be tall? Hopefully you see here that every single box contains a dominant allele, which means every offspring will be tall. So 4 out of 4 is 1 times 100% would be 100% of the offspring are expected to be tall. So for the assignment, you're going to need to complete several of these genetic problems. Remember to follow the same set of rules. Establish your parent genotypes, set up your Punnett square, fill it out to determine the answer to the question.